All right, in this chapter, we're going to talk about the regulation of gene expression. In this chapter, specifically, we're only going to cover section 13.3, gene mutations. So we've already talked about gene expression and how the genes in DNA are transcribed and translated into proteins, or the physical representation of that genetic information. But now we're going to discuss what happens when this process goes awry and causes genetic mutations. A genetic mutation is a permanent change in the sequence of bases in DNA, and the effect of the gene mutation can range from no effect at all to having a complete inactivity of the gene. There are two types of mutations. There are germline mutations. Germline mutations occur in sex cells and can be passed down from one generation to the next. There are also going to be somatic mutations. Somatic mutations occur in the body, or body cells or somatic cells. These are not going to be passed down through the generations. They typically only affect a region of the body. But they can be fatal. Just because they can't be passed down doesn't mean that they are not fatal. Cancer is a great example of this. Some mutations are spontaneous, meaning that they appear to happen for no reason, whereas others can come from environmental influences. So spontaneous gene mutations result from abnormalities in normal biological processes. Remember when we talked about the proofreading mechanism of DNA polymerase? DNA polymerase not only polymerizes DNA, so it synthesizes new DNA, but it can also proofread and correct any mistakes that it makes. However, sometimes DNA polymerase may miss a mistake. There may be an error in the copying, which can lead to a mutation arising in the DNA genome. There are also induced mutations. Induced mutations are caused by environmental factors, things like exposure to toxic chemicals or radiation. So like I said, spontaneous mutations can be associated with a number of different processes. These are typically chemical changes in DNA that lead to a mispairing during replication. Or there might be short movable segments of DNA called transposons that can be switched from one chromosome to another, or they can actually slide around and move within the chromosome itself and cause mutations that way. There's also the possibility of spontaneous mutation arising from replication errors. Like I said, DNA polymerase is responsible for proofreading the DNA as it's being synthesized. When DNA polymerase, when it proofreads as it goes, it is still possible to make a mistake. The error rate for DNA polymerase is about 1 in a billion. Induced mutations are, by their very definition, have to be induced or caused by something. Mutagens are the term that we use for environmental factors that cause mutations to arise, such as radiation or organic chemicals. Many mutations that are induced are often going to be somatic mutations. These types of mutations occur in a small area in the body after the person is born, but they may lead to cancer. So these types of mutagens are also called carcinogens for their cancer-causing properties. Some of the environmental mutagens are UV radiation. Too much unprotected exposure to sunlight can cause cancer. Tobacco smoke. Tobacco smoke contains multiple carcinogens, and it has been linked to 1/3 of all cancer-related deaths. So how can scientists determine if a chemical is mutagenic? A lot of times we're going to use what is known as the Ames Test for mutagenicity. This is where we take a particular type of bacteria that requires an essential nutrient to survive, and we grow them in the absence of that nutrient. So for the Ames test, we're going to use a bacteria that requires histidine. They don't have it themselves, they require it to grow. We're going to grow them in the absence of histidine. So we're going to put them on a plate, and we're going to put them on a petri dish that lacks histidine. Then we're going to expose our test group to the mutagen. If the suspected mutagen is able to mutate the bacteria DNA, so we expose them to that chemical mutagen, And if we then grow them on the bacterial, on the petri dish that does not have histidine, if the bacteria are able to grow, if they have mutated to the point where they no longer need histidine to be provided for them, we can see that a mutation has happened. And so the material that we're using does have mutagenic properties. There are a variety of mutations that occur that can ultimately affect protein activity. One such type is what's known as a point mutation. Now, it's called a point mutation because it involves a change in a single DNA nucleotide. The change is going to alter transcription and can potentially lead to a change in the amino acid sequences produced by translation. This type of point mutation is sometimes called a base substitution, where one incorrect nucleotide is substituted for the correct nucleotide. This can lead to disastrous effects if this change of the amino acid encoded by this new codon, or it could potentially result in a stop codon that truncates the protein early. Or there is the potential that nothing will change, and there will be no need for concern. There are a lot of codons that will all code for the same amino acid. We have a lot of redundancy built into this system to help protect us against point mutations. Here's an example of what that could look like. Oops. If we had a single point mutation right here, where this A gets swapped out for a G. Pardon me. If we had a single point mutation where this A gets swapped out for a G, we could see the replacement of phenylalanine with leucine. So the original amino acid sequence is methionine, phenylalanine, threonine, arginine, isoleucine. Now the sequence reads methionine, leucine, threonine, arginine, isoleucine. Now this isn't a big deal if phenylalanine and leucine have a lot of the same properties. As we kind of talked about before, it is the characteristic properties of the amino acids that ultimately control the shape and the structure of the protein. If we substitute out one hydrophobic amino acid for another hydrophobic amino acid, especially if that one hydrophobic amino acid is nonpolar, we may not see much of a change. The protein might, be shaped roughly the same way it was always going to be shaped. If that protein wasn't the active site of an enzyme, the protein might be okay, and it might be able to kind of limp along and do its job. But if we substitute out a base, if we substitute out a nucleotide base that has completely different properties than the one we needed, we could see a complete disruption of that protein's activities and the complete inactivation of that protein as a whole. So that's why point shift mutations can have some interesting effects. They may not affect it at all. They may lead to drastic and serious consequences for the protein as a whole. There are also frameshift mutations. Frameshift mutations occur a little bit differently. Frameshift mutations are caused by when one or two nucleotides are going to be deleted, inserted or deleted from the DNA. This is always going to render the protein completely non-functional. And the reason why is because, if you remember, we talked about our codons. Our codons are going to be 3 nucleotide sequences. They're always going to be read in 3 nucleotide sequences. If we were to delete a letter, we would completely shift the frame. So, for example, here is the sentence that's made up of 3-letter words. The cat ate the rat. If we were to accidentally delete this C, it would change the entire frame. The first 3 letters, the, are still there, but the next 3 letters are A-T-A. The 3 letters after that are T-E-T. Then we have H-E-R and then A-T. So we have completely destroyed the sentence. If we change one letter, we're going to shift the codons by one place, and our sentence, which used to read the cat ate the rat, now reads the at-a-tet her at, which is complete nonsense. It makes no sense. It is not a protein that we want. If we were to accidentally insert another nucleotide, so we were to insert another C in this location, again, that would shift the frame completely, and we would now have a sentence that reads the cat ate a rat. We would completely change the meaning of the sentence. So here's a little bit of what that looks like. Our original sequence was methionine, phenylalanine, threonine, arginine, and isoleucine. If we were to add a single nucleotide, we would suddenly have a sequence that reads methionine, phenylalanine, aspargine, leucine, aspargine. And I know that doesn't look like nonsense, or it looks like just as much nonsense as the original sequence, but this is a complete nonsense. It's a non-protein. It's not going to function the way that we want it to. It's not going to behave the way we want it to, because the entire amino acid sequence has been botched. If we were to delete a nucleotide, the same thing would happen. If we were to delete a single nucleotide, we deleted that single T. We would suddenly have a sequence that reads methionine, leucine, arginine, glutamate. So we're going to see a complete change in the appropriate amino acid sequence. So what are the consequences of having a faulty protein, or a protein that's not functioning the way it's supposed to? Well, it kind of depends on what its job was. But if a faulty enzyme is inserted into a metabolic pathway, a person may be unable to convert one molecule to another molecule without serious consequences. So just for example, in the disorder phenylketonuria, PKU, phenylalanine is going to build up in the system, and the excess can cause an intellectual disability. In this case, if we are missing the protein that is responsible for converting phenylalanine to tyrosine in order to produce melanin, which is just your skin pigment, we're going to see a dangerous buildup of phenylalanine in the bloodstream. It's not just that the skin will be very, very pale, but that we can see caused intellectual disability caused by that buildup of phenylalanine in the bloodstream. Another great example of this, so that would be, we could have, if there's a non-functional protein in a metabolic pathway, we could see a buildup of proteins. We could also get insensitive receptor proteins. So for a great example of this, androgen insensitivity. With androgen insensitivity, cells are unable to respond to testosterone due to a faulty receptor for testosterone. So if we do not have the androgen receptor, it does not matter how much testosterone the body produces. The body is not going to respond to that testosterone. It's not going to pick it up. So female instead of male genitals will form, and female instead of male secondary sex characteristics are going to occur at puberty. In other words, the individual will be genetically male. They'll be an XY male, but without the androgen receptor molecule, they will present as female. They will appear female because they will not be able to respond to the testosterone produced by their body. They will not be able to produce the male sex characteristics or the male secondary sex characteristics at puberty. There are also mutations. The development of cancer involves a series of accumulating mutations that can differ from one type of cancer to the next. So the tumor suppressor genes, remember when we talked about tumor suppressor genes, they're responsible for inhibiting cell division. If we take those out, or if we see mutations in those tumor suppressor genes, it's like ripping the brakes out of your car. We don't have the ability to stop cellular division if we have a mutation in our tumor suppressor genes. Proto-oncogenes, on the other hand, stimulate cellular division. Proto-oncogenes stimulate cellular division. Mutated proto-oncogenes can become oncogenes that are always active. Mutations in oncogene and tumor suppressor genes stimulate the cell cycle uncontrollably and can lead to tumor formation. All right, and that's it for this chapter. I'm going to let you read through this summary slide on your own. Please make sure that you review everything, and please let me know if you have any questions, comments, or concerns.